Hello students. In the last video we have started the vectors. The first part of the vector we have completed, where I have given the characteristics of the vector and the different sites. So the four sites we have discussed. What are they? They are first site that must be present in one vector, that is the OD. Second one we have discussed the ROP. Okay, and the third one we have discussed, that is selectable marker, and the last one, fourth, that was we have completed in the last video, that is the recognition site or the cloning site. So these four parts we have discussed. The next part, fifth part, we need to know, that is the vectors for the cloning genes in plant and animal host cell. So just see, there is a vector that is known as a natural vector. Okay, so there is one natural vector which have the capacity, okay, it has the capacity to transfer the genes into the host body naturally. Okay, we don't have to do anything, or it is not genetically modified, or it is not purposely inserted, but it has this characteristics that there is one pest present, one actually pathogenic organism is there, that is known as Agrobacterium. Okay, Agrobacterium tumefaciens. Okay, so remember this name. This is actually one pathogen of the dicot plants. Okay, it's a pathogen of dicot plant. Okay, so in case of the dicot plants, this Agrobacterium tumefaciens in the root region actually they affect, and it transfers one gene. That gene is known as the T gene or the Ti plasmid. Okay, Ti plasmid stands for the tumor inducing plasmid into the dicot plant. So that's after that what happens. Suppose you just take this example. This is the plant, right? Now what happened? This is the root. Okay, now this Agrobacterium tumefaciens, this is one pathogen. Okay, here the pathogen is there. Suppose this is the pathogen. So in this pathogen they have one gene. That gene is known as the Ti gene, or that is also known as the Ti plasmid 2. Now, whenever that root of the dicot plant is affected by this Agrobacterium tumefaciens, this pathogen, then what happens? This root is also have the root cells, that means the host cell. So this Agrobacterium tumefaciens, this have this characteristics that it can naturally transform this Ti plasmid into the root. Okay, naturally. It is not genetically modified, it is not genetically inducing, but this Ti plasmid is naturally can be transferred by this Agrobacterium tumefaciens into the dicot plant root, right? So whenever it is inserted, what will happen? That this plant will start to develop the cancerous growth or abnormal root growth. Tumor type structures will start to grow. Okay, so whenever all those tumors grow, actually this Agrobacterium tumefaciens will be also be beneficial. Why they are transferring this Ti plasmid into the root? Because these root cells which are now affected by this Agrobacterium tumefaciens, they will start to release some chemicals which is needed for this Agrobacterium tumefaciens. Okay, so just remember that this natural vector Agrobacterium tumefaciens, it is going to have one very unique characteristics, that it can transfer. Remember, natural vector that is Agrobacterium tumefaciens, this is one pathogen of the dicot plant which transfer the Ti plasmid, okay, or tDNA. Okay, what that T stands for? T for the tumor and I for inducing. So tumor inducing plasmid is able to naturally transfer from the Agrobacterium tumefaciens body to the host. Okay, so the host will develop a tumor and induce to produce chemicals necessary for Agrobacterium tumefaciens. Okay, so what is that? That will be the characteristics. You need to know that one vector which is present, which is also known as the natural vector. Actually, from here also the concept have been developed that yes, some of the vectors can transfer the genes, right? So that genes can be transferred from one vector to the host body, so that some changes can develop in the host body. Now this host body which can develop the tumor, and whenever they're developing the tumor, those tumor affect, or they're having the tumor, so tumor cells of the host, they can produce some chemicals, which that chemical actually is needed by the Agrobacterium tumefaciens. Remember that this is very much important thing. From here one question comes, and what is that question? What is the natural vector? That is the Agrobacterium tumefaciens, who transfer the TI or the tDNA or TI plasmid naturally from the body to the root cells, so that the root cells can develop the tumor, and they are directed to produce some chemicals necessary for the Agrobacterium tumefaciens. Okay, so that's all about the characteristics you need to know about the vector. So these are all about the vectors. After that, let us see the examples of the vectors. After that, let us see some examples of the vectors, right? So the first vector, which is a natural vector, and the second vector, which is first artificial vector we are going to discuss. So the first vector is the plasmid, right? So this plasmid is the natural vector which is extracted from the bacteria, which is one double-stranded, okay, just see, this is one double-stranded DNA vector which is extra chromosomal. Extra chromosomal means it is not necessary for the bacteria's own metabolism. Even if we remove that, the bacteria can survive. So this is extra chromosomal circular double-stranded DNA molecule present in some bacteria. Now what is that extra chromosomal means actually? Okay, so it is not required for the normal bacterial metabolism, but it is important as it has the antibiotic resistant gene. Okay, that means it gives the bacteria to survive in some type of antibiotic like ampicillin or tetracycline. So if the ampicillin or tetracycline is given, the bacteria will not survive. This plasmid will give that bacteria a resistance, so that even in the antibacterial medium, that is ampicillin medium or tetracycline medium, this plasmid, because of this plasmid, the bacteria survive. So that's why this plasmid is necessary. Okay, it gives a little bit strength or immunity to the antibacterial. Now the next one, to the different type of antibiotics. Okay, so see that next one, it can be single or double or multiple copied plasmid. Multiple copied means it can produce one copy, it can produce two copy, or it can produce around 200 copies too. But in general, 200 copy, if it can produce, we can take that one only. Yeah, so but most common is the multiple copied plasmid, which is the most commonly used natural plasmid. Okay, it is just extracted and used for the different purposes. But for the first time, the scientists Bolivar and Roderick developed the first artificial plasmid. Okay, so this is the first artificial plasmid, that is the PBr 322. So the first PBr 322, what actually is, what that PBr stands for, see, it is the first artificial vector. Remember that, this is very much important. Okay, now it is constructed by the two scientists Bolivar and Roderick. Okay, there are two scientists Bolivar and Roderick. According to their names, this plasmid has given the name. So what is that P stands for? P stands for plasmid, B stands for Bolivar, R stands for Roderick, and 322 is the sample number. So see here, P, that stands for plasmid, B for Bolivar, R for Roderick. Now we need to know that it has 8 restriction enzyme recognition site. That means 8 different type of genes can be inserted. But the thing is that all of these 8 genes or 8 recognition sites cannot be cut by one enzyme, only one enzyme. Okay, suppose we are giving only one specific enzyme. Suppose one restriction enzyme we have added, then only one specific region will be cut. So for 8 restriction recognition sites, they have to be utilized 8 different type of restriction endonuclease. For one restriction endonuclease, one only recognition sites will be cut. Okay, so already I have given here, suppose Hind tree will cut only in this region. Okay, so this will be a recognition site. BNH1, this is one recognition site. SL1, this is one recognition site. PST, this is one recognition site. PVU, CLA, these are all recognition site. Okay, that means in these specific regions we can just add the specific enzymes, restriction enzyme, like suppose Hind tree, then that specific regions can be cut. We can insert our gene of interest. So this is the restriction enzymes recognition sites. It has 8 restriction enzyme recognition sites, but for one restriction enzyme only one recognition sites will be present. Okay, now next one, they are going to have only one OD, then ROP and 2 selectable markers. So what are the 2 selectable markers? They are ampicillin resistant gene and tetracycline resistant gene. Now what is that meaning? That in this ampicillin medium also this plasmid will survive. So because of that, the host within which that PBR322 is there, it will be survived. Or in the tetracycline medium, which is also one antibiotic, the bacteria will die. And because of this PBR322, as it is going to have the tetracycline resistant genes also, they are going to survive. So that's why it is a very important thing that because of the selectable marker, we can at least find out which is the the reason by which we can able to find the recombinant from the non-recombinant. Suppose we have a number of bacteria within which we have inserted PBR322. So whenever we have inserted the PBR322, that PBR322 will have this ampicillin and tetracycline resistant gene, right? So because of that, in this suppose a different type of bacteria which is normally growing, suppose they are not going to have this ampicillin site. Okay, so if you add the ampicillin medium, then the transformant is going to survive, and the non-transformant which don't have the PBR322, that will not survive. Okay, so by this selectable marker we can find out the recombinant from the non-recombinant or not, isn't that? So that's why what we can call this PBR322, which is actually one highly used artificial plasmid actually, so that vector which can actually help in transferring the different genes, because in different genes we can cut the different sites and we can add different gene fragments. Okay, and the desired gene fragments will be inserted into the host body. Okay, so that is a 2 type, but otherwise a number of other vectors are also here. Let's just see. After that, the next third type of vector, let's just see what is that. PUC, plasmid. So what is the P stands for? P stands for plasmid, U for university, C for California. So University of California, they developed the plasmid, where actually one specific type of selectable marker for the first time was used, and that was the selectable marker LegZ gene. Okay, so there are 2 type of selectable markers present. One is the enzyme producing gene, that is the enzyme producing gene is the LegZ gene, which is producing one type of enzyme, that is beta galactosidase, which actually will be used in finding or to separating the transformant from the non-transformant. And second one is the antibiotic resistant gene. So here a specific type of selectable marker is used. What is that? That is the LegZ gene. Remember that LegZ gene is an enzyme producing gene which produces one enzyme, that is beta galactosidase. Okay, so beta-galactosidase is produced by the lexatzine. So this is one selectable marker, which is the enzyme producing gene that is useful for the finding out or to separate the transformant from the non-transformant. Now the next type, that is known as a TI plasmid. Already I have told you that from the Agrobacterium tumefaciens, the pathogen itself can transform the gene, that is TI or the DNA, into the root of the plant so that they develop the tumor and also produce the chemicals. This is necessary for the Agrobacterium tumefaciens. So this is one natural plasmid, actually natural vector, which have the capacity to transform the gene fragment into the host body. Next one is the RI plasmid. So RI plasmid is extracted from the Agrobacterium rhizosome. Okay, so same type of like the TI plasmid obtained from the Agrobacterium tumefaciens. Similarly, RI plasmid is extracted or obtained from the Agrobacterium rhizosome. Okay, the next type, there are different type of viruses. So a number of viruses are here which help in the transformation of the gene fragment, different type of gene fragment, DNA, single-stranded, double-stranded, different type of DNA into the host body. So let's just see some of the vectors. They are the viruses or the phages. First one is the lambda phage. So lambda phage is used to carry double-stranded DNA. Next one, M13 virus. Remember that M13 virus, which is useful for carrying or transforming or chain to carry actually single-stranded DNA. Then retrovirus, which is used to carry the DNA into human. Next one, another type of vector is also here. That is the cosmid. Cosmid is the artificial vector, okay, produced by adding cos gene of the virus to the plasmid. Next one, we have phagemid 2. Phagemid is produced by adding gene of the bacteriophage into the plasmid. Same thing like cosmid and phagemid, similar type of vectors. They are used in transforming, adding the gene of the bacteriophage to the plasmid. The next one is the actually two type that are 9 and the 10, YAC and BAC. YAC stands for yeast artificial chromosome and BAC stands for bacterial artificial chromosome. So these two chromosomes, actually plasmids or the vectors, they were actually developed, formed artificially in the Human Genome Project in US. Okay, in Human Genome Project already you will see we will know that we have one project that is known as the Human Genome Project carried by US, and they actually found how many genes they are present in our body, in our cell. In each cell, how many genes we have? We have around 30,000 genes. Earlier it was thought that around millions of genes were present, but surprisingly only 30,000 genes we have, which is responsible for developing all those characteristics. So for this whole process, Human Genome Project, to find out how many genes we have, okay, so for that different type of vectors were actually synthesized artificially in the laboratory to carry actually large large molecular weight DNA segments into host that are developed during Human Genome Project HGP to carry the large DNA fragments. So these are all about the different type of vectors. Most important vector, remember only two vectors, they are going to be very much important. One is the artificial and another is the natural. So natural one is the TI plasmid. Remember that this is going to be very much important. TI plasmid, from where it is extracted, how it is working, everything will be needed. And another one is the artificial vector, which is PBR 322. Plasmid developed by Bolivar and Rodrigue, which is the first artificial type of vector, which is actually going to have 8 recognition site, 1 or 8 2 selectable markers. They are the tetracycline and ampicillin resistant genes. So that 2 are going to be very much important, but otherwise also if you want to remember you can, but it will be not needed for the board's exam, but for the NEET you can see all of these examples. But yes, that PBR 322 diagram will be also necessary. So diagram is given already in the NCERT. You just find out all the characteristics. Okay, remember their names as well as the artificial and natural. That's all about the factors. We have completed the factor part. In the next video we will see continue the next part. So that's all about it. I hope you understand. Thank you.